Assalamualaikum, my name is Nur Batrisha binti Ghazali. My metric number is 201722. I am from Group 8 that will cover a topic which is model system aging to study the process of aging and triggers for aging. Group 8 members are Siti Nur Suhaida, Nur Aisyah, Nur Aini and Nur Amira Wahida. I will be the first presenter. What is aging actually? Aging is progressive physiological changes in an organism that lead to senescence or a decline of biological functions. It is a process that goes on over the entire adult lifespan of any living thing. It takes place in a cell, an organ, or the total organism with the passage of time. There is a field that studies the aging process which is gerontology. Gerontology is devoted to the understanding and control of all factors contributing to the finitude of individual life. There are several theories that propose the reasons why aging process occur. The first one is gene regulation theory of aging. This theory proposes that senescence, which is in Malay is penuaan, is the result of changes occurring in the gene expression. In other words, lifespan is largely determined by the genes that we inherit. Next is telomere theory of aging. Telomeres are specialized DNA sequences that located at the ends of eukaryotic chromosomes. When a cell divides, the telomeres become shorter and shorter each time until they are gone. At this point, DNA cannot be copied anymore, and the cell simply ages and is no longer able to replicate. This is due to the absence of the enzyme telomerase, which is normally expressed only in germ cell, which is in testis or ovary, and in certain adult stem cells.
Okay, next is about process of aging. So basically, the exact mechanism are poorly understood, but recently scientists have identified non-physiological traits ranging from generic changes to alteration in cells' regenerative ability that play a central role in aging. The 9 hallmarks of aging are altered intercellular communication, telomerase attrition, cellular senescence, mitochondrial dysfunction, deregulated nutrient sensing, epigenetic alteration, stem cell exhaustion, loss of proteostasis, and genomic instability. So as the years pass, our bodies accumulate genetic damage in the form of DNA lesion. This occurs naturally when the body's DNA replicates. Mitochondria are especially prone to this damage. Mitochondria produce ATP, regulate many different cells' activity, and also plays an important role in programmed cell death. If mitochondrial function decline, then cell and later on whole organ deteriorate too. Other changes are known to occur in expression pattern of gene, also known as epigenetic alteration, that affects the body tissue and cell. Gene silenced or expressed only at low level in newborns become prominent in older people, leading to the development of degenerative diseases like Alzheimer's, which accelerate aging. Next, cellular regeneration decline as we age. The DNA in our cell is packaged within chromosomes, each of which has telomeres. Telomeres shorten every time cells replicate. So when the telomeres are too short, cells stop replicating and die, slowing the body's ability to renew itself. With age, cells increasingly grow senescent too, a process that halts the cell cycle in time of freeze, like when the cancer cells are proliferating. But the response kicks in more as we age, halting cell growth and cutting short their ability to replicate. Aging also involves stem cells that reside in many tissues which have the property of dividing without limit to replenish other cells. As we get older, stem cells decrease in number and tend to lose their regenerative potential, affecting tissue renewal and maintenance of our organs' original function. Other changes revolve around the cell's ability to function properly. As they age, they stop being able to do quality control on protein, causing the accumulation of damaged and potentially toxic nutrients, leading to excessive metabolic activity that could be fatal for them. Lastly, intercellular communication also slows, ultimately undermining the body functionals.
Hi, my name is Nur Aisha, metric number 201726. I will be explaining the triggers of aging, which includes biological, lifestyle, and psychological factors. Biological factors occur biologically in the individual and difficult to alter, such as genetic, metabolism, and oxidation, wear and tear, and glycation in the body. This is the main triggers of aging, which is genetic, as mentioned by Suhaida. I want to add that dysfunction and build-up of protein can also trigger aging process, as it is responsible for regulation in aging and growth in the body. Lifestyle factors are factors that are affected by lifestyle of a person, like addiction to substance, eating behavior, and so on. These factors are not the main triggers but may contribute to premature aging. First is diet, because inflammation in the body can contribute to aging process. Next, alcohol and caffeine intake. Drinking alcohol excessively dehydrates your body, causing the skin to sag and lose its shape. Caffeine may have similar effect, although there's conflicting research about the daily coffee consumption cause wrinkles. Next, toxin in cigarette smoke expose your skin to oxidative stress, and this cause dryness, wrinkle, and other signs of premature aging. Tanning bed and exposure to sun penetrate the skin with UV rays. These rays damage the DNA in your skin cell, causing wrinkles. Sleep gives your body opportunity to refresh and regenerate cells. Thus, sleep disturbance can increase signs of premature aging as well. Finally, is psychological factors. Stress hormone and inflammation can age our body faster. It is also found that people who have suffered from eating disorder also tend to develop wrinkles faster. Other than this, environmental factors could also trigger premature aging, but not as much. That is all from me. Thank you.
Assalamualaikum and hi everyone. My name is Amira and I'm going to explain the relationship between animal tissue culture and aging model system. So first, how do scientists actually study aging model system? So scientists use a tissue engineered construct to control the cellular level damage and cell-cell distance in individual tissues to establish the role of complex interdependence and interactions between cells in aging tissues. So usually scientists will use a synthetic tissue. Okay, so second, what is actually synthetic tissues? So synthetic tissues are materials designed to substitute for natural tissues, and they even exhibit enhanced properties. Okay, so next, why does scientists actually use tissue culture to study aging? So first, because it is not possible to actually control the cellular level aging characteristic and cell-cell interaction strength in vivo tissues. And plus, tissue engineering techniques allow for direct tests on synthetic live tissues. So these structures are sufficiently complex and controllable that we can explore the hypothetical idea about the nature of aging. So next, what is actually the outcome of studying aging model system using tissue culture? So one of the outcomes of the study is to quantify the interaction between cells that lead to tissue level failure. So we found that while microscopic mechanism drive aging, the interdependency between cells play a major role in tissue death, providing evidence on how cellular aging is connected to its higher systemic consequences. So organisms die not because they run out of cells, but rather due to complex systemic problems. Thus, a complete theory of aging must go beyond cellular damage. In this work, scientists experimentally verify the elements of this theory with the use of synthetic tissues and demonstrate for the first time the systemic spread of failures from cellular level to tissue level, and propose a fresh view of systemic aging. Okay, so from what I've explained, scientists actually study the hierarchical spread of failure from cells to tissue by growing synthetic tissue in well-controlled hydrogel microenvironment, in which they may vary intercellular distance, environmental stress, and also the age of cells. Okay, so lastly, what are the findings about aging using tissue engineered construct? So the findings actually support that aging cannot be solely explained by failures of individual cells, but it is an emergent phenomenon involving strong intercellular interaction. So first, we actually find that systemic aging is more important factor than cellular aging. So this means that a healthy young cell is more likely to die if its neighbors malfunction compared to an old or stressed cells with intact neighbors. So the second finding is we find that cellular aging is tightly coupled to systemic aging. So since aging the cellular level causes cells to lose their ability to interact with surrounding cells, specifically we determined that one of the causes underlying systemic aging is the loss of ability to receive or internally process functional cooperative factors from surrounding cells.
Hello, my name is Nur Aini binti Nazri, and I'm going to present the limitation of aging model system. So the first one is it is nearly impossible to tell whether an organism is representative of human aging or not. It has been argued that some aging mechanisms got public are common to all species, while others are unique, which is private of each species. Certain animals appear to age for different causes than us. At present, it is impossible to tell for sure if a given model of organism is accurate or how accurate it is. The second is laboratory strains are often genetically homogeneous because laboratory strains tend to provide more consistent results, but also gives rise to discrepancies between strains on the effects of genes or interventions on aging. The third one is human physiology can be much different than that of model organisms. To give an example, the phenotypes of aging in yeast and humans are totally unlike. Not even researchers working on yeast expect yeast aging to be a perfect mirror of human aging. It is possible that some general pathways of aging are conserved between all organisms, but certainly not all age-related changes overlap. So the fourth one is the telomeres. The telomeres. Clearly, they are major players in cellular senescence in human cells as well as other organisms. Telomeres deficient mice are normal up to 4 generations and then show some signs of accelerated aging. On the other hand, telomerase dysfunction in humans causes a disease called dyskeratosis congenita, which shares some features with the 6 generations. With the 6 generations telomerase deficient mice. It is therefore nearly impossible to determine the impact of the telomeres and telomerase in human aging, based on model system. Even between mice and humans, there are similarities but also differences in the biological outcomes of telomerase deficiency. The last one is all model organisms are considerably shorter-lived than humans and were developed for laboratories research based on their high fatality. Not only this means the different evolutionary processes acted on these organisms and on humans, but selection and fatality may have also selected for short lifespans in laboratory strains that generate biases in aging studies. In other words, it has been argued that the life-extending gene variants found in these organisms may be simply restoring lifespan to what is normally found in the wild. The fact that wild-derived mouse strains take longer to reach sexual maturity and live significantly longer than common laboratory strains supports this view.
To sum up, the study of aging is undoubtedly not a simple one. This is because aging is a normal phenomenon process, but advancing our fundamental knowledge of aging, it can promote the understanding of age-related disease. Thus, further studies are still needed and numerous cues need to be solved. Next slide is the list of the references that our group has used. That's all from our group. Thank you.